Good morning, student. Today we are going to start exercise number 15, that is designs used in plant breeding experiment. So student, this exercise is so much important because whatever in future you will do a trials in your MSc, this exercise will help you to carry out your experiment. So this is important exercise for your future education also. So this exercise will be practically implemented by you in your master course. So first of all, we will see our terminologies which are required to learn this exercise. First one is a experimental design. What is meant by experimental design? So this is the various types of plot arrangement. It is a different type of plot arrangements are there, and these various type of plot arrangement which are used to test a set of treatment. What is meant by treatments here? The treatments means it is dose of fertilizers or dose of pesticides or any thing which we have to do a comparison among these. It is called as a set of treatment. So various arrangement of plot arrangements, various types of plot arrangements which are used to test a set of treatments. Okay, the plots are made to which are used to apply the different treatments. If I have one variety of soybean, that is JS335, and on that variety I have to study the effect of different doses of fertilizer. So these doses of fertilizer, different doses of fertilizers are the set of treatments. Okay, so for which purpose we do a different arrangement of the plots to test the set of treatment to draw valid conclusion is called as experimental design. So in short, it is the various arrangement of the plots, okay, which is used to test the set of treatment to draw valid conclusion is called as experimental design. Then what is meant by experiment? Okay, so the definition of experiment is a scientifically planned method is called as experiment. Okay, those methods which are scientifically planned, these methods are called as experiment. Then what is meant by treatments? The various objects of comparison is called as treatments. Okay, so various objects of comparison. Okay, if I have a variety of soybean, that is JS335, and on that variety, if I want to study effect of different doses of fertilizer on that variety, then the different doses of fertilizers are the treatments. Okay, so those things which we have to study the different doses, or in which we have to do a comparison. Okay, so various objects of comparison is called as treatments here. Then experimental unit means on which we will apply a different treatments, then it is called as a experimental unit. So JS335 is the experimental unit on which I have to study different doses of fertilizer. So what is meant by the treatments? Treatments are the various objects of comparison is called as treatments. Then experimental unit. Experimental unit is the JS335, the group of material to which a treatment is applied in a single trial experiment is called as experimental unit, on which we apply, on which things we apply a treatment. Those basic material is called as experimental unit. If we take a example of patient, okay, different age, if we take an example of patient, then on a group of patient, the different medicines, different doses of medicine. So doses of medicines are the treatment, and the experimental unit will be the patient. Okay, then next part is that the experimental error. So the error, error means the whatever the variation due to the environmental factors or uncontrolled factors is called as experimental error. Okay, for that purpose, if I took two plots and if I gave a good favorable environmental condition to that plot and the stress condition to that plant to that plot, and if I plant a same variety here, okay, the variety is the soybean variety JS335. If I apply on these two plots, okay, but the condition is that for A plot I am applying a good favorable condition, and for B plot I am not applying whatever the good condition favorable conditions there, I will give a stress to that plot, but whatever the material I have planted in these two plot are the same. So what happened exactly? The A plot of the same varieties gives more yield than that of the B plot. Okay. So what happened here? Here the variation occurs in a same genotype. So such type of the variation when on a piece of land, if I have a piece of land, okay. So whatever the fertility of that soil present here is not same as that of the here, or is not same as that of the here. So the variations will occurs there. The variation in plant performance of this place is different than that of this place, is different than that of the this place. So whatever the variation occurs due to the environmental factors or those factors which are uncontrolled factors are there, like fertility or the environmental variations are there. So what the variation which occurs due to the environmental factors and uncontrolled factors is called as experimental error. Okay, because the error or the variation in that variety which is purely due to the environment or which is due to the uncontrolled factors, so it is called as an experimental error. Okay, so so this is so much important, and our main motto to learn experimental design is that to reduce environmental error. If we reduce experimental error, then variety the whatever the varieties are there, it get um, genotypically get expressed. If there is a less influence of the environment, then the variety express its genotype more. Okay, so whatever the expression of that variety is, it is only due to the hereditary material, it is not due to the environment. And if the variations or the if the very differences into the same variety is due to the environmental condition, then that variation will not pass from one generation to next generation. Only the heritable variations will pass from one generation to next generation. So our main motto to learn experimental design is to reduce the experimental error. So we plan the arrangement of the different plants like in a such way that due to which the whatever the experimental error will occur there get minimized. So it may be the arrangement differently. There are the many different plot arrangement are there. So we will learn in upcoming slides. Okay. So there are the three principles. First, there are the three principles to design experimental design. Okay. So the three principles of experimental design. First one is randomization. Next one is replication, and the last one is local control or the error control. Okay, error control means what? The whatever here is our motto is that to reduce the experimental error. Okay, the process by which we reduce the experimental error is called as the error control. Okay, whatever the error is occurring in our experiment, that we have to reduce there by a different mean, then it is called as a error control. So first principle of experimental design, it is not in your manual, so write down these points, that is principles of experimental design. And if you read your manual after my teaching, then it is somewhat difficult to understand you. So I have prepared my PPT in a such way that you can easily understand the different experimental design, and whatever the things I have discussed in the different experimental design, for that these terminologies are so much important, because randomization, replication, local control I will use in this presentation again and again. So you should understand what is meant by the randomization. First one is randomization. What is meant by randomization? So let us take first example. This is the first why there is a need to take a replication and randomization. See here, this is the plot. This is the plot here, and here I have made a three subplots there. Okay, it is called as a replication. So what is the definition of replication? The repetition of the treatments under the investigation is called as replication. First of all, what is randomization? Okay, so here what happened, the block, the name is given. Firstly we take a one example, that this is the piece of land here, and if I plant, if I made such a way that if I made such a parts of that land to plant 5 varieties, so what happen in this case, the whatever the fertility is present in that area is obviously different than that of the fertility of the land which is experienced by the genotype 5. So ultimately there is a differences may occurs in genotype 1, 2, 3, 4, 5, and we cannot drawn a valid conclusion there, because the soil fertility is not same in all the 5 genotypes is not getting the same soil fertility there. Okay, so there is a need to do a randomization here. Okay, firstly, so I am here using somewhat Marathi to for your easy understanding. मला या ठिकाणी काय म्हणायचंय, जर माझं एक शेत अशा पद्धतीचा एक शेतामध्ये प्लॉट आहे आणि मला 5 जीनोटाइप लावायचे, आणि मी त्या शेतामध्ये 5 प्लॉट अशा पद्धतीने पाडले आणि पहिली व्हरायटी अशी लावली त्या प्लॉट मध्ये, तर काय होत आहे, पहिल्या व्हरायटीला मिळणारी जी सॉइल आहे ही ऑब्वियसली 5 नंबरच्या व्हरायटीला मिळणाऱ्या सॉइल पेक्षा काय आहे, डिफरंट आहे. इट मे बी अ मोर फर्टाइल. सो ज्या ठिकाणी ज्या व्हरायटीला मोर फर्टाइल सॉइल मिळालेली आहे, त्याचा ऑब्वियसली परफॉर्मन्स कसा असणार आहे, हाय असणार आहे. पण त्या ठिकाणी अशी पण शक्यता असू शकते, जर तुम्ही इथे इमॅजिन केले की हाय फर्टिलिटी आहे सॉइल, आणि इकडे जसं जसं तुम्ही पास होत आहात तसं फर्टिलिटी ही लो होत चाललेली आहे, तर मग काय व्हायले की ऑब्वियसली 5 नंबरची जी व्हरायटी आहे लेस परफॉर्मन्स देणार आहे दॅन दॅट ऑफ द फर्स्ट व्हरायटी. सो आपण कंपॅरिझन या 5 व्हरायटीचं करत असताना व्हॅलिड कन्क्लुजन काढू शकत नाहीये. ओके, जर अनदर केस मध्ये जर तुम्ही 5 इथं लावली असती तर मे बी तिला 5 नंबरच्या व्हरायटीला हाय फर्टाइल सॉइल मिळाली असती आणि 1 नंबरच्या सॉइलचा लो परफॉर्मन्स आला असता. म्हणजे हे जे एरर येत आहे, हे जे व्हेरिएशन येत आहे, इट इज ओनली ड्यू टू द सॉइल फर्टिलिटी ऑर द एन्व्हायरमेंटल फॅक्टर्स ऑर अनकंट्रोल्ड फॅक्टर आहे. मग याला जर आपल्याला रिड्यूस करायचं असेल तर आपण एक काम करू शकतो, रँडमायझेशन करू शकतो. पहिले तर हे जे 3 प्लॉट रँडमायझेशन, आता या ठिकाणी बघा, या ठिकाणी आता हा प्लॉट विसरून जा. मग मी काय करणार की अशा पद्धतीने माझ्या व्हरायटीची अरेंजमेंट करणार की तिला कधीतरी हाय फर्टिलिटी, कधीतरी मीडियम फर्टिलिटी, कधीतरी लो फर्टिलिटी मिळाली पाहिजे. मग त्यांचा ऍव्हरेज घेतल्यानंतर मी त्याच्यावरून काय काढू शकते, व्हॅलिड कन्क्लुजन काढू शकते. मग जर तुम्ही या ठिकाणी पाहिलं तर बघा, 3 नंबरचे जीनोटाइप इथं दिलं, त्याच्यानंतर 3 नंबरच्या जीनोटाइपला ह्या टाइपची एक सॉइल मिळाली आणि एक लो फर्टिलिटीची सॉइल मिळाली. ओके, मग याच्यामुळे काय जे एरर होत होतं, जे मीडियम फर्टिलिटीला लावल्यामुळे आपण त्याचा एक स्टेबल परफॉर्मन्स मिळू लागला, ज्याला की हाय फर्टिलिटी सॉइल पण मिळत नव्हती, लो फर्टिलिटी सॉइल पण मिळत नव्हती. पण अशा 3 ठिकाणी 3 नंबरचं जीनोटाइप लावल्यामुळे काय झालं, त्याला लो, हाय, मीडियम तिन्ही फर्टिलिटीला त्यांनी त्याचा परफॉर्मन्स दिलेला आहे. मग याच्यामुळे जे ह्या अरेंजमेंटमुळे जे हाय लेवलचं एरर येत होतं, ते एरर आपण या ठिकाणी काय करू शकलेलो आहे, मिनिमाइज करू शकलेलो आहे. ओके, सो what is meant by randomization? It is the allocation of treatment. Here are the 5 are the treatments. Whatever the varieties we used here, these are the treatments here. So Randomization means it is the allocation of treatment of a different plots by a random process. So here whatever the here I am there is no same format are there. Okay, firstly what I do here, मी काय करणार, ह्या मोठ्या प्लॉटला एकदा 3 प्लॉट मध्ये अशा आडव्या लाईन्स मध्ये डिवाइड करून घेणार आणि नंतर त्याचे प्रत्येकी 5 प्लॉट पडणार, असे 5 त्रिक 15 प्लॉट पडले. मग व्हरायटीचं अलोकेशन करत असताना मी असं काही पर्टिक्युलर सिक्वेन्स डोक्यात न ठेवता रॅन्डमली अलॉट करणार प्लॉटला ती ट्रीटमेंट, याला म्हणतात रॅन्डमायझेशन. मग या ठिकाणी 3 5 1 4 2 किंवा तुम्ही घेऊ शकता 2 3 4 असं काही पण घेऊ शकता. परत दुसऱ्या रिप्लिकेशनला देत असताना तुम्ही काय करणार, या ठिकाणी परत एकदा रॅन्डमली प्रोसेस देणार. फक्त केअर अशी घ्यायची की 2 सेम व्हरायटीज एका जवळ एक येऊ नये. तर या ठिकाणी हे एक जे आलेले आहे ते आपण या ठिकाणी टाकू शकलो असतो, 5 या ठिकाणी सुद्धा टाकू शकलो असतो. म्हणजे रॅन्डमली काय केले जातं, अलोकेशन ऑफ ट्रीटमेंट टू द डिफरंट प्लॉट्स बाय रॅन्डम प्रोसेसनी जे अलोकेशन केलं जातं त्याला आपण रॅन्डमायझेशन म्हणतो. त्यानंतर आहे रिप्लिकेशन. The repetition of the treatments under investigation is called as replication. It is the repetition. काय केलेलं आहे बघा, हे जे आहे आता हा जो आडवा प्लॉट पडलेला आहे ज्याच्यामध्ये एक ट्रीटमेंट ही एकदाच ऑकर होत असते, हा पूर्ण जो मोठा प्लॉट आहे याला आपण म्हणत असतो रिप्लिकेशन. ओके, it is the replication. So the repetition of the treatment under the investigation is called as a replication. So here the repetition in each replication occurs only once. ओके, परत दुसऱ्या रिप्लिकेशन मध्ये परत एकदा ते ऑकर व्हायले आणि तिसऱ्या रिप्लिकेशन मध्ये परत त्याचा एक रिप्लिका फॉर्म व्हायलाय. तर हे जे रिपिटेशन ऑफ ट्रीटमेंट आहे त्या प्लॉट मधलं ब्लॉक मधलं, तर त्याला आपण काय म्हणत असतो, रिप्लिकेशन म्हणत असतो. त्याच्यानंतर आहे लोकल कंट्रोल ऑर द एरर कंट्रोल. द प्रिन्सिपल यूज ऑफ ग्रेटर होमोजिनिटी. या ठिकाणी आपल्याला काय करायचे, हे प्लॉट्स बनवून आपण त्या व्हरायटीला या ठिकाणी होमोजिनियट सॉइल फर्टिलिटी द्यायलो, किंवा या ठिकाणी तुम्ही 2 नंबरच्या व्हरायटीचा जर विचार केला फक्त त्या ब्लॉक पुरता, तर त्याला त्याच्यापुरती एक होमोजिनियस सॉइल मिळालेली आहे. ओके, सो द प्रिन्सिपल यूज ऑफ ग्रेटर होमोजिनिटी इन द ग्रुप ऑफ एक्सपेरिमेंटल युनिट फॉर reducing experimental error is called as a local control. Here our main motto is to reduce the experimental error. So how we can reduce the experimental error? By the use of greater homogeneity. Okay, so what is meant by randomization? Randomization is the allocation of treatment to the different plots by the random process. Here, see here, T1 T5, but only the condition is that in each replication the one treatment will appear only once. Okay, so here only once it will appear in this replication. Then what is meant by replication? Replication is the repetition of the treatment. It is the repetition of the treatment is called as replication. So how many replications are there? Because the here every treatment is repeated 3 times, so 3 replications are present there. For replication, the another word we can use which is called as a blocking, or simply it is called as a block. And there is a greater use of homogeneity is used, and by this we reduce experimental error. Whatever the error is creating here due to the soil fertility, that will be ultimately reduced in this case. So there is a greater use of homogeneity and it reduces the experimental error. It is called as local control or the error control. So these are the 3 principles of the experimental design. Okay, so next slide is about how to select a single plant selection. If you observe this piece of land, the piece of land showing the average productivity here, and the above soil showing the above average productivity, and this soil showing the below average productivity. So how to select a single plant from such a land? Okay, so whenever you select a single plant from this area, then it will give a more productivity. If you select a plant from this area, then it will give a more productivity. So how we can reduce the error which is created due to the variation in the soil fertility or the productivity? So there are the 2 methods. First one is the grid method. In grid method, what happened in this grid method, the grid method is given by Gardner. This is important for your competitive examination also. And what they do there, okay, they make a grid. They subdivide a main plot into the small plots, and within each plot, if you only things forgot these grids, okay, forgot these grids, so what happened, the fertility of soil of this area is definitely different than that of this area. If I compare this plant with this plant, then it is not fair. Okay, so what the gardener do there, they form a grids there, or they subdivide the big plot into the subplots. Okay, and then they do a comparison of these subplots with each other. Whatever the plants present here, they compare this plant with this plant. So whatever the error creating while comparing this plant with this plant get minimized. Okay, so by this method you can select a single plant from that particular plots also. The next method of selecting single plant here is the honeycomb design, which is given by Factorial in 1973. Here, every plant in the population is evaluated by placing it in the hexagon. So whatever the here are the some hexagons are there. So each of the plant is here. It is compared with whatever the plants which are the present within that hexagon. Okay, the plant is only compared whatever the plants are present in that hexagon. Then again this hexagon is moved. Okay, again this hexagon is moved to the another place. Okay, by moving hexagon, every plant is compared with a different group of plants in the population. So what happening here, this plant is compared with that whatever the plants which are present on that edges of the hexagon. But when you move this hexagon here, whatever the plant is here, it is compared with this plant. So every time whatever the plants you place into that hexagon is compared with other group of the plants. So the whatever the comparison you are going to do here is the correct one, because every time you are comparing this plant with the every other plant. If you took this hexagon, then the group of compared plants are the different here. So the another one reason why it is called as honeycomb, because whatever the structure for comparing these plants you made there with the help of hexagon is looks like a honeycomb design, so it is called as a honeycomb design. Okay, so the simply the one hexagon is formed there, then this hexagon is placed on that plant, and whatever the plant present inside that, it is compared with the edges of that the plants which are present on that edges. Okay, or on that angles of that hexagon. Then again this hexagon is moved on another plant, and this plant is compared with the other group of the plant. Okay, so this is the honeycomb design which is given by Factorial. Then next design, which is so much important now, we will interested, these are the two designs which are used for the single plant selection, and the next are the designs used for the replicated test. So first one, the method or the design which is used for the replicated test. Replication means what? It is the repetition of the treatments, and here if you observe here the repetition, the treatments are repeated here, the E, then again here is E, and again here is E. Okay, or if you observe here the red color, positive control, positive control, positive control, these 8 times this red color is repeated there. So the repetition 8 times, so these are the designs which are for the replicated data. Okay, so the first design is completely randomized design. It is also called as CRD design. Okay, so first sentence for about the CRD is, it is suitable for a laboratory experiment or where the material is highly homogeneous. See here student, this is the CRD method is only used for the pot culture or the laboratory materials, or the whatever the different strains of the cultures of the bacteria or the fungi. For that purpose it is used, the completely randomized design is used. So what happened here, the whatever the material you have selected, or the whatever the soil present there in plots, these are highly homogeneous. Okay, so the experimental error is not operated here. Okay, you should understand that in CRD, the experimental error, the error due to the soil fertility or the error due to the environmental factors are not operating there, because because you are giving a control condition into a laboratory. So whatever the environmental errors are there, it get eliminated, and whatever the soils you are using to fill the pots, it is highly homogeneous for all the pots. So the material is highly homogeneous, so there will not be any error due to the environment. So the error control is not operated in this design. Okay, so the error control is not operated, and this is mostly used for the homogeneous material in laboratory purpose. Then also it is used for the pot culture. There is no restriction on number of treatments. There is no restriction on randomization and the replication. Okay, you can, if you have 4 treatments, then as like A, B, C, D, then you can repeat this. You can give a 3 replication for A treatment, then for B treatment you can give a 2 replication, for C treatment you can give a 5 replication, or for D treatment you can give a 3 replication. So there is no restriction. It is a non-restricted method. So it is called as a non-restricted method, because here there is no foundation to apply the number of replication to the treatments. Okay, and the next one is, there is a no any thumb rule for the randomization also. You can, in a one replication, in a one line, you can take this one treatment again and again. So there is a no such thumb rule for that randomization. So it is the only method which is a non-restricted method, because there is a no foundation on how many number of replication you have to use, and how to do the randomization here. And third one thing which is important for your my objective point of view is that the error control is not operated here, because whatever the material you are using, by drawing a valid conclusion in the CRD, that is a highly homogeneous material. So whatever the error due to the variation is not operating here, and so error control is not operated here. There is no restriction on number of replication and the number of and the process of randomization here. This is the only method which is used for the pot culture and the laboratory purpose. Okay, so this is the completely randomized design. Then next design is randomized block design. It is called as RBD. So here is my cursor. Okay, so this design, the short form of this design is RBD. Okay, randomized block design. So homogeneous unit is called as blocks here. Okay, whatever the homogeneous units are made here, these homogeneous units are called as block here. Okay, the field is divided into equal number of replications. The field is divided into number of replication. The number of replication is R1. It is indicated by Roman numbers. Then R2. The field is divided into equal number of replications. The field is divided into equal number of replications. So 4 replications are there, and each replication is again divided into equal size of plots. Okay, these are the equal size of the plots are here. The size of each block, or the size of each, it is called as a block also. So whatever the plots are formed here, it is also called as a block, and the size of each block is equal, and the size of each replication is also equal. So if you consider this replication, the size of each replication is same that of the other replication, and the size of each block is same. So this design control the fertility variation in one direction. So if you have a field of in one direction only, in one direction only, it controls the fertility direction. So if you have a land of rectangular size, then the fertility of this portion is obviously different than that of the this portion. So it minimize the fertility variation in one direction only. Okay, so this is important for your examination, that this design controls the fertility variation in one direction. Then variability within the blocks, the whatever the variability present in within the blocks is less than that of the between the blocks. There may be a variability, there may be a more variability within the blocks, because here you are planting 10 number of 10 number of genotype, and here you are planting 8 number of genotype. So whatever the variability within between the blocks are more than that of the within the blocks. So in within the blocks only the 5 number of plants of 5 number of genotypes are there. So within the block the variability is less than that of the between the blocks. The error, how here the error is controlled by making a homogeneous replications, and by making a homogeneous block, whatever the error is creating here is minimized here. So how the error is controlled here? By making a number of replication, by making a replication, and by making a blocks, the error is controlled in that design. So in next practical we will see how to draw valid conclusion by using randomized block design. The whatever the problem is there, the 1 hour problem is there, the problem is so much big, but so easy problem is on that RBD analysis. In next practical we will see a RBD analysis, that how to draw a valid conclusion if RBD data is provided to you. So this is important. So what points we covered here: the field is divided into a number equal number of replication, then each replication is divided into a block, then whatever the variation present within the blocks is less than that of the between the blocks, then error is controlled by making a homogeneous by making a equal number of replications, and by making equal number of blocks. Okay, so this is a these are the this is all about the randomized block design. So I am using eraser here, and I will erase this, because this is important to this is important for you to draw this diagram for your practical assignment. In practical assignment also you have to draw this diagram of RBD. Then next one is factorial randomized block design. So here if here for example the one, if one genotype is here and on that genotype 2 doses of fertilizer is studied there, so it is called as factorial RBD. Then in that there is a 2 plots are made for factor 1 and factor 2. So this is the factorial RBD. Okay. The effect of number of different factors are investigated simultaneously here. So it is called as factorial RBD, that is FRBD design. Then next one design is a Lattice design. Okay, students, so observe here first. This is the piece of land. Okay, this is the piece of land. So in that piece, what they do? They firstly made a columns and the rows there. Okay, so in LSD design it controls how in the RBD yeah, RBD only so in case of RBD what happen, it controls the fertility in one direction only. But here in case of Latin square design we are forming a rows here and also the columns here. So these controls the, due to the columns, due to the rows, what happened there, it controls the fertility of the horizontal soil, and due to the forming of columns it controls the fertility vertically. So it controls the fertility in two directions. Okay, it is important for your objective point of view that the LSD controls the fertility variation in two directions. So what they do, they gives a number to the columns and then they gives a number to the rows. only the allocation, how the allocation is done there. The A, B, C, D, E are the genotypes there. So how they assign genotypes to the different plots. Firstly, they allot one genotype in a such way that it will occur only once in a row and only once in a column. If you observe here, the B only once occurs in that row and also in a column. Then if you take an example of B here, then that B will occur only once in a row and only once in a column. Okay. If you take an example of that D, then that D is also occurs only once in a row and only once in a column. Okay. So the assignment, the treatment are assigned in a such way that the each treatment appears only once in a row and only once in a column. This is important for you. And the number of plots in this, it is equal to the square of treatments. Number of plots, how many plots will form there? Obviously, how many number of rows will be occurred there? If we take a 5 genotypes, then in every row, then it form a 5 rows and 5 column. So it will 5 as a 25. 25 plots will be formed there. So the number of plots is equal to the square of varieties. And how we are controlling errors here? By forming a rows and column of the plot. By forming a rows and column of the plot, we are controlling the fertility or the variation due to the fertility in two directions. So by forming rows and columns here, we can easily control the experimental error which is occurred due to the environmental factor or due to the uncontrolled factors. Okay, so this is the Latin square design. Then next plot is a split plot design. So what happens, student, here? If you see here, in case of split plot design, there are three types of the plots are there. First one is a main plot, then subplot, and then ultimate plot is there. So in case of split plot design, the plot is divided into main plot, subplot, and the ultimate plot. In this design, the several factors studied at the same time. We can study the different plots at the same time here. The factors are such that the sum of them requires a large area. So there are the many factors such as the irrigation or the depth of flowing or the sowing date. So these factors requires a large area to study, and those factors such as fertilizer, then method of pruning, then name of different varieties, then these requires a small area. So whatever the factors are there, those factors which requires more area, this is allocated to the main plot first. Okay, then these main plots is subdivided into the the main plot is divided, and the plot which result, it is called as a subplot. Again, when we divide this subplot, then it get converted into the ultimate plot. So here, if there are three factors are there, then those factor, out of three factors, if we consider A factor, then B factor, and the C factor. If A factor is requiring more area, then it is allocated to the main plot first, then main plot is again divided, then the B factor is allocated to this plot, and at the last, those factor requiring less area, then this C factor is allocated to this by dividing this subplot, it is called as a ultimate plot. So this is the arrangement of the split plot design, where the two or more factors can be studied at the same time. So firstly, we have to divide a field into a main plot, then this main plot is again divided into a subplot, then subplot again divided into a ultimate plot. So how here the error is controlled? The error is controlled by making a homogeneous block. So for it is only for your understanding, students. These things are only for your understanding, that how they draw these main plots and subplots and the ultimate plot. So actual design of this split plot design is likewise. Okay, again they divide this main plot into the subplot, main plot into a subplot, then again that subplot is again get divided likewise. So the final structure what we observed here will looks like. Okay, so this is for your understanding. They only draw the to understand what is meant by main plot. So what is the main plot here? Okay, this is the main plot. Okay, then what is the subplot here? So whatever the subplot is here, these are the subplots. Okay, so this is for your understanding. And how the error is controlled here, by making homogeneous blocks here. Whatever the they made a homogeneous blocks here, and they can minimize whatever the error which is arising due to the fertility of soil. So in split plot design, the experimental plot is divided into main plot, subplot, ultimate plot. In this design, the several factors can be studied at the same time, like the irrigation, depth of flowing, sowing date, then fertilizer dose, then methods of running. Then those factors which are requiring large area, these are allocated to the main plot first, and then the other factors are allocated which requires the smaller area, then these are allocated to the small plots. The each block is divided to the number of first factor. Each subplot is divided into subplots by the second factor, and which is equal to the second factor, and each subplot is divided into ultimate plot, which is equal to the third factor. So whatever the ultimate plots, the number of ultimate plots is always equal to the number of third factor. The number of subplots are equal to the number of second factor, and number of first plot is main plot is equal to the number of first factor. So this is about the split plot design. Then next one is the lattice design. Here the student, it is in incomplete block design. This design only we can use where the square shape of the field is available. In this design, the number of varieties always form a square. If you observe here, in each replication, the number of varieties, how many varieties are present here? 25. So this number of varieties is forming a square here, that is 5, 5, 25. So this is the incomplete block design. In this design, the number of varieties form a square, and each block is divided in a such way that the number of plots in blocks form a square. So how many the square shape will be formed there? Okay, they form a square there. So the size of the blocks are square, and also the size of replication is also square. There is a no restriction on number of replication. You can take there is no replication, you can take 4, 5, 6, the even number of replication you have to put there. So this is the incomplete block design, and this design can be used where the variety form a squares like 16, like 36, then 25, then 49, then 81 or 64. So here the one first criteria is that here the number of whatever the number of genotypes or whatever the number of treatments are there, it must form a square there. Okay, so this is all about the lattice design. Then next one is a augmented design. In case of augmented design, the augmented design concept given by Federer in 1956, and this design can be used where we have to screen large number of germplasm lines, or large number of whatever the genotypes on a limited area. So here whatever the checks are there, these checks only replicated, and cultures are not replicated. So whatever the capital letters are shown here, these are the checks here. So the only the checks are repeated, ABCD, 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 and whatever the germplasm lines indicated here, these are of the small letters. So the germplasm lines are not replicated here. E, F, G, H, I, J, K, L, M, N, O, P, Q, R, S. So the whatever the germplasm lines are there, they are not replicated, and whatever the checks are used to draw a valid conclusion there, or to screen a germplasm line, only the checks are replicated there. So this is the augmented design, and you have to draw this diagram of the augmented design also. Okay, so this is all about your different experimental design, and this is the assignment on exercise number this is the assignment on exercise number 15, that is draw diagrammatic layout of CRD, RVD, LSD, split plot design, lattice design, and the augmented experimental design. So again we will review this experiment because it is somewhat different than your earlier syllabus. So in this experiment we see what is meant by the experimental design. It is the various plot arrangement. Then what is the experiment? It is scientifically planned method is called as experiment. Then what is meant by treatment? Various objects of comparison is called as treatment. And what is the experimental unit? The group of material to which the treatments are applied is called as the experimental unit. And what is meant by the experimental error? The variation due to the environmental factors or the uncontrolled factors is called as experimental error. And our main motto of experimental design is to reduce the experimental error. Okay, by making a replication, or by making a homogeneous block, or making a rows, by making a columns, by making a main plot, subplot, ultimate plots. In that way we can reduce the error. And only one design is there where the local control is not operated, that is CRD, because whatever the material we use in the CRD, it is highly homogeneous. So these are the principles of the experimental design. First one is randomization. What is meant by randomization? It is the allocation of the treatment to the different plots by a random process is called as randomization. What is meant by replication? It is the repetition of the treatment under the investigation is called as a replication. And what is meant by the local control? The principal use of greater homogeneity in the group of experimental unit for reducing experimental error is called as a local control. So whatever the errors are occurring in the experiment, that error we have to reduce to draw a valid conclusion. So it is called as a local control, or it is called as a error control. Error control which is caused due to the environmental factor or uncontrolled factors. So so first design is a CRD. CRD is operated where the material is homogeneous, or it is used in the laboratory, and it is also used in the pot culture where material is highly homogeneous. Material is highly homogeneous, so there is no matter to create a variation there. So error control is not operated here. Then it is a non-restrictive method because there is no restriction to on number of replication and the process of randomization that we use. Then randomized block design. Here the homogeneous blocks are formed. The variation within a block is minimum, and the variation between the blocks are more. Then it controls the fertility variation in one direction, and the error is controlled by making an equal size of replications and equal size of blocks. Then Latin square design. The Latin square design is where the rows, the field, it controls the fertility variation in two direction by making rows and the columns. They assign each genotype in a such way that it must be occurs only once in a row and only once in a column. Okay, so this is a Latin square design. Here also the number of plots is always equal to the number of varieties. Okay, so now how many number of plots will occur there is equal to the square of that number of genotypes. Error are controlled by making homogeneous by making the rows and columns. Then next is split plot design. Here the several factors we can study at the same time. Those factors which require some large area that is allocated to the main plot, then main plot divided into subplot to which the second factor is applied, and the third factor is the ultimate plot where how many number of ultimate plots are there, that the third is equal to the third factor. So here by making a homogeneous block we can reduce the error here. Then lattice design. Lattice design is an incomplete block design where the varieties form a square and each block divided in a such way that the plots in each block form a square, and there is no restriction on number of replication in case of lattice design. Then next one is the augmented design. The augmented design is given by Federer in 1956. Here this design is used to screen a large number of germplasm on a limited area. So whatever the checks we use there, that only the checks are replicated here, and whatever the germplasm lines are there, they are not replicated. And last one is the assignment for you on exercise number 15, that is draw diagrammatic layout of completely randomized design, randomized block design, Latin square design, split plot design, lattice design, and the augmented design. Only you have to draw diagrams of these different designs here. Thank you.